So it's really a great pleasure for me to be introducing Dr. John O'Shea. Um, Dr. John O'Shea did his undergrad at the St. Lawrence University, and he then went on to finish his medical degree at the University of Cincinnati, did his residency at sunny upstate New York, and then went to the NIH to do his postdoctoral fellowship. And since 1994, John has been at the NIH. He is now the director of the Intramural Research Program at the National Institute of Arthritis and Musculoskeletal and Skin Diseases. So it's really a great pleasure to have Dr. O'Shea here with us today, where he is going to share with us some of his recent work. And since his lab started in 1994 at NIH, John has made, and his colleagues has made lots of seminal discoveries, including the first cloning of the tyrosine kinase JAK3 and its important role in the signaling cascade for interleukin-2. And since then, I think John and his members of his lab has made lots of important fundamental discoveries, examining the role of the STAT family of protein transcription factors that, as we all know, are really important in the immune system in particular. And of course, John has received many, many awards, so I won't be naming each one, but John has been named the NIH Directors Award for 4 times, as well as the AAI Steinman Award for Human Immunology. And in 2014, he was elected to the National Academy of Medicine. And I'm sure our trainees will also be really interested to talking to John later this afternoon, because John is also on the editorial board for Immunity and JXMed. So hopefully our trainees will be getting plenty of advice from John on how to publish. And I think today John is bringing us a really French title, looking at cytokine signaling, and proves that Sean, the more things change, the more they stay the same. I'm sure we're going to look forward to your seminar. And with this, let's welcome Dr. John O'Shea for our last seminar of the Immunology series this year. Welcome, John.
Thanks so much. Thanks. I'm going to share my screen, see if I can get this to work. And let's see, I got it to work when we practiced. Let's see if I can do it now. And let's see, can you see my screen? And let's see, I'll get it on the right. And let's see, let me get my pointer to work. Okay, everything look good, and you can hear me okay and everything?
Yes, looks great, and we can hear you perfect. Thank you, John.
That's great. And I can see some of you are keeping your cameras on, which is nice, because in these sort of Zooms, you sometimes wonder, am I talking to outer space or something? So Wendy, good to see at least one person there, and you can make faces if I'm not making any sense. Okay, so I want to try to get in 4 quick stories if I can. And I want to start with a translational story, which I think may interest you, but then sort of switch to the more things change, the more they stay the same, and thinking about how we think about cytokine signaling in the 21st century, and in particular focusing on these complex regions of the genome we call super enhancers, et cetera. And I'll give you 2 examples of that. So just starting, as Wendy said, way back in a previous century, when I was trying to get tenure, the problem that faced us was, how do cytokines act on their receptors and regulate gene expression? And that problem really was solved by a number of groups working together, but largely from the work of Jim Darnell and George Stark, et cetera, where this pathway, which at first seemed pretty perplexing as to how cytokines can rapidly turn on gene expression, they do it in a very efficient way that's preserved between mammals and insects, and even to a certain extent down to dictyostelium. And everybody's seen this. cartoon in various textbooks. But when we cloned JAK3, as when he mentioned, and identified JAK3 as patients with X-linked SCID and then JAK3 SCID, Warren Leonard and I proposed the idea that if you purposely targeted JAKs, you might have a new class of immunosuppressive drugs. And so 20 plus years later, that's where we are now. We have 9 approved JAK inhibitors shown here for various indications, starting with the first JAK inhibitor approved was ruxolitinib for polycythemia vera, where there's constitutive activation, gain of function mutations of JAK2. But the drug that I've worked with most in collaboration with Pfizer is tofacitinib, which our original idea was that it would be a JAK3 selective inhibitor, but it turns out to inhibit JAK1, JAK3, and also JAK2. But now that's approved for rheumatoid arthritis, psoriatic arthritis, juvenile arthritis, including remarkably for children from age 2 and up, ulcerative colitis. The third JAK inhibitor approved is baricitinib, approved for rheumatoid arthritis, approved for atopic dermatitis in Europe, and again, remarkably approved for the use of COVID cytokine storm, and maybe we can talk a little bit about that later. Bunch of other inhibitors approved. I'm not going to go through all of them. The one inhibitor I always have to mention, of course, every time I give a talk, people usually point out that their dogs are on JAK inhibitors, and there is a faculty member, because of HIPAA, I won't disclose the faculty member at UCSD whose dogs are on JAK inhibitors, but in fact, the canine JAK inhibitor oclacitinib is approved for atopic dermatitis and really revealed even before JAK inhibitors were used in people the efficacy of this drug in this indication. But I wanted to talk about a small story, and that's the use of JAK inhibitors in lupus, just very quickly, and more to illustrate a couple of points that I think are sort of compelling. So when you think about a disease like lupus, systemic autoimmune disease, there are lots of reasons why you could think that JAK inhibitors might be useful. And thinking about that, you think of all of, or I think of all of the cytokines that are exert their effect by the JAK-STAT pathway. And obviously things like interferons are prominent in a disease like lupus, but there are also other cytokines as well that contribute to pathogenesis. And so lots of reasons why you might think that JAK inhibitors might be useful in this disease. And in fact, we did preclinical trials, as you would suspect, and the drugs did turn out to be efficacious in that setting. But equally, there's a lot of reasons why you might think that JAK inhibitors could be problematic in a disease like lupus. And one issue, of course, is that the first generation JAK inhibitors affect multiple JAKs, including JAK2. And so the use of JAK inhibitors can be associated with cytopenias. And that's a problem in lupus, which is already associated with cytopenias. So that's one issue that you might imagine could be problematic in using these drugs in this disease. But the other issue is that now that we have better drugs for lupus, the real cause of morbidity and mortality of lupus is myocardial infarction, cardiovascular disease. And JAK inhibitors do increase cholesterol, and there's a fair amount of discussion as to whether they also can increase cardiovascular risk. And in a study that's completed but not quite published yet, but which the FDA pointed out in a trial in which JAK inhibitors and TNF inhibitors were used side by side in rheumatoid arthritis, that the risk of cardiovascular disease was increased somewhat in patients on JAK inhibitors versus TNF inhibitors. And the other thing, theoretically, and it sort of is humorous in a way, when I was collaborating with Pfizer, back in the like I said, in the late 20th century, thinking about cytokines and blocking their effect, we didn't know about Tregs at the time, so we didn't know about the role of IL-2 in generating regulatory T cells, because FOXP3 hadn't been discovered yet. And so that, in thinking about a drug like tofacitinib that can block JAK3, one might imagine that you could actually make autoimmune disease worse. So for all of those reasons, we thought we would do a sort of phase 1-ish trial in this disease. But there was another thing that we were thinking about, and that was, thanks to Dan Kastner and other investigators' work, that everyone knows, of course, that autoimmune disease is associated with MHC. That's the genetic link. But surprisingly, one of the biggest links GWAS hits in lupus and rheumatoid arthritis, Sjogren's, is a polymorphism associated with STAT4. And so that, needless to say, got our attention. And what we did in this small trial, which we did at the NIH, was we actually took subjects who did and did not have the STAT4 risk allele, and we're trying to figure out, would that make a difference if you gave patients JAK inhibitors. Now, I have to say, this definitely wasn't my idea. I said, no, we're never going to see a difference. I guess I thought that the difference in subjects who did not have this risk allele would be tiny. I mean, don't forget, there are a lot of people around who have this risk allele and don't have lupus. But in any case, I was outvoted. And as I'll show you momentarily, that's a really good thing that the group outvoted me. Betty Diamond in particular said, no, I think there'll be a difference, even in this small trial with 30 patients. So what we did, and here's the group here with Mariana Kaplan and Safraz Hasni really leading the team, and Betty Diamond I mentioned. We did, the obvious thing was just check to see, do you see alteration in interferon signature? And you can see on the top are subjects who got the drug during the period of the trial, and you can see reduction in the interferon signature, these various genes that I'm sure are similar to or very typical interferon inducible genes are reduced in subjects who are on the drug. And then here are various controls, patients on placebo, and then what happens to the interferon signature after the trial is terminated. And you can see that the interferon signature goes down here. And the other thing that Mariana Kaplan is very interested in the role of neutrophils in lupus, and one of the things she measures quite well is the generation of neutrophil nets, netosis. And this is a very prominent aspect of the immunopathology in lupus. And remarkably, what was found is that patients who have the STAT4 risk allele, they actually have more netosis than subjects who are STAT4 risk allele negative. And what happens is when you then treat these patients with JAK inhibitors, you can see that you have a more pronounced effect on patients who have the STAT4 risk allele. Not shown are the controls, but you see you get a significant decrease in neutrophil nets on subjects who have this risk allele. And again, it was less prominent in subjects who did not have the STAT4 risk allele. So the other thing that I mentioned was, what is the impact of JAK inhibitors in this cohort on cardiovascular sort of aspects of the disease? And what you can see here is that, in fact, in these subjects who got tofacitinib, you see increased cholesterol, but it's HDL cholesterol, the so-called good cholesterol. And what you see associated with that is increased cholesterol efflux. And so there's a preferential increase of HDL with no change in LDL or triglycerides, increased LCAT. And along with this, Mariana Kaplan had non-invasive vascular function performed. And in fact, along with the increased HDL, there was actually improvement in vascular stiffness and the like. So just because I want to move on to some other newer stuff, I'll just summarize this. And so what we found is that there were no unexpected adverse events. The adverse events were what we would expect from having seen thousands of patients on these drugs. We saw inhibition of the interferon signature, which is what we were hoping to see as sort of a positive control. But the other part is that really this, to my knowledge, this is the first trial that stratifies subjects with the use of JAK inhibitors by the STAT4 risk allele. And that was notable to us because, in fact, you saw increased netosis in these patients, and these are the ones who had a response to tofacitinib. What we saw in terms of cardiovascular aspects is that we saw increased cholesterol, but it was increased HDL cholesterol. And again, we saw a more prominent effect in subjects who had this STAT4 risk allele. So our hope going forward is that there are plenty of trials going on, but we hope that people take this into consideration, that if you're going to use a JAK inhibitor, that it might be a good idea to assess the cohort in advance so that you have a significant number of patients who have the STAT4 risk allele. We did this purposely in setting up the trial in advance, but certainly going forward, we would think that this would be an important aspect of trials. There is a trial that has been reported with baricitinib, which again showed efficacy and no safety signals. There are ongoing trials with deucravacitinib, which is, I'll touch on in a moment, is a particularly interesting compound because it's a selective TYK2 inhibitor, and given the role of interferons and the like in lupus, this is an interesting drug to keep your eyes on. As I mentioned, there's an FDA statement saying that cardiovascular risk are greater in our patients on tofacitinib versus TNF inhibitors. But in our work, it suggests, but is by no means definitive, that actually you may get a benefit in lupus patients. So whether this is disease specific or other factors, but it's certainly something that needs a lot more work. And thinking about cardiovascular risk with tofacitinib or not, by no means that's not the biggest paradox with this class of drugs, is that we knew from the earliest days that if you were treating patients with JAK inhibitors, that infections might be a risk. But you know, the last year seeing the utility and emergency use authorization of baricitinib for COVID, where actually you can reduce mortality and complications with COVID septic shock, that or COVID cytokine storm, was just a tremendous surprise. And now we just recently, there's a trial that was reported from Brazil using tofacitinib for COVID-19. So that's a sort of a very long-winded way of saying we're just beginning to understand JAK inhibitors and how they work in patients in different settings. So I'll just give a quick summary of where we are with JAK inhibitors. So the first class of JAK inhibitors inhibited multiple JAKs, and as a result would target basically almost all of the 57 cytokines that use the JAK-STAT pathway. But we're now in the phase where we have various selective JAK inhibitors. Selective JAK inhibitors have now been approved, upadacitinib, filgotinib in Europe, and other agents. But you can see that selective JAK1 inhibitors are going to target a smaller spectrum of cytokines than the first generation JAK inhibitors. Although I'd point out that by selective, for the clinicians in the audience, selective is really, I mean, relative selectivity. So upadacitinib, for example, especially at higher doses, will have some activity towards JAK2. But we also have JAK2 and TYK2 inhibitors coming along. We have selective TYK2 inhibitors. I mentioned the BMS compound, which is interesting because it actually targets the pseudokinase domain of JAKs, which in principle can make these drugs even more selective. We have selective JAK2 inhibitors and then selective JAK3 inhibitors. So lots of ongoing trials in these areas. I'm not going to go through all of these details here, but everything from, you know, as I mentioned, COVID to different forms of arthritis, interferonopathies, including Down syndrome, and other autoimmune diseases, including alopecia areata, a lot of activity here. to both leukemias, lymphomas, and solid organ cancers. But I would say, you know, this is maybe a different lecture, but I just want to make the pitch that there's so much more work to be done with this class of drugs. It's really, you know, steroids were discovered and first used in the 1940s and 50s, but obviously for many years we have been trying to figure out exactly what's the best way to use steroids, and it took many years to do that. And to a certain extent, we still don't know exactly how we're going to use steroids and JAK inhibitors in combination. What's the best way to do this? What are we going to measure? What aspects of disease are we going to use the drugs? Obviously, some of that is ongoing presently with COVID-19. Dexamethasone does have efficacy in cytokine storm, but how do you use this or not with JAK inhibitors? Anyway, more questions than answers, and I'll move on to the next part, which is gives the title of this of my talk, which is the more things change, the more they stay the same. And I think that came from a Bon Jovi album, or maybe a French philosopher. I can't remember which, but in any case, what I wanted to move to for the rest of the talk is thinking we had these cartoons that we generated in the 20th century, and the idea really was that thinking about how transcription factors bound to promoters. And at the time, of course, we didn't have the human genome and all of its complexity, only to find that only 2% of the genome was genes. And as you know, in San Diego better than I know, that there's all the rest of this genome here, and importantly, it's not junk. It's active in different ways in different cells, in different contexts, and not only that we have transcription throughout the genome that it goes way beyond the canonical genes that we enumerated in the human genome. But that you can look at activity of the genome by looking at thousands of cell-specific enhancers using different techniques, ChIP-seq and ATAC-seq, et cetera, and trying to understand how all of this relates to regulation of genes and the regulation of the transcription of non-canonical genes. How does that relate to gene expression and the behavior of cells? So I wanted to go through that a little bit, and maybe again go back about 10 years for us. And when we first started doing these sort of experiments, we were pleased and surprised to see that in T cells, when you polarize cells to Th1 and Th2, there are about 20,000 active enhancers, and about 50% are shared between Th1 and Th2 cells in this very simplistic reductionist experiment that we did in vitro. But what we also found is that there was almost no sharing of active enhancers when we looked at Th1 cells, Th2 cells, macrophages, and embryonic stem cells. And that was puzzling first, but I'll show you what is, in a way, the resolution of this seeming paradox. And the one thing we found at the time was a major role of signal-dependent transcription factors, and to a lesser extent, in the way we did this experiment, lineage-defining transcription factors. And I'll be returning to this in a bit. And of course, the other thing we found out is that enhancers are not distributed sort of symmetrically throughout the genome. There are regions of the genome that has this high density of enhancers, and this is referred to as super enhancers or stretch enhancers by Francis Collins and Rick Young in the opposite order in which I said that. And where these enhancers are enumerated is through K27 modifications or P300 ChIP-seq. These super enhancer regions are associated with GWAS hits. And the idea that Rick Young pitched is that super enhancer is associated with cell identity. And when we did this in immune cells, we found that super enhancers are associated with genes encoding cytokines, cytokine receptors, transcription factors, and also long non-coding RNA. And so I'll return to this idea of identity versus activation momentarily. And so for the next bit of the talk, I'll be sort of discussing, why do we have these super enhancers? Although I don't really know the answer to that. Why do you have these complex enhancers, and what's the relationship to chromatin accessibility? And this work was really started by Hanyu Shi, and then she continued it in the next story that I'm going to tell. And now Hanyu has her own lab at the NIH. And Hanyu noted in thinking about innate and adaptive lymphocytes, which both have the capacity to have selective gene expression, especially production of cytokines like type 1 cytokines, interferon gamma, IL-4, IL-13, etc. That what happens in innate cells is that you have this stepwise accessibility that occurs in a developmental manner, so that even in early in their development, innate cells develop accessibility of effector loci, but that's not what happens in T cells. T cells go through their development, and naive T cells really, these key loci are largely inaccessible, and what happens is during activation, these regions become accessible, but you more or less end up in the same place, that these loci become accessible in very similar manners, but the mechanisms by which this occurs is different. But we thought, wait a minute, you also activate these cells. So what would activation look like in innate cells? And so that's what Hanyu wanted to pursue. And we were thinking about this problem here, which again, loads of people, including Chris, have thought about this over the years, and Giacchino Natoli as well, about how you could have condensed chromatin, that pioneer factors act on these regions, open the chromatin, and then signal-dependent transcription factors come and drive gene expression. And so the pioneer factors really established competence for the signal-dependent factors. So in that sense, you might argue that pioneering factors create cell identity and then permit the action of the signal-dependent transcription factors. And so we wanted to see whether this really, this model system related to what was going on in NK cells. And as I'll get to in a moment, the answer is yes and no, as sort of at the same time. And I'll explain what I mean by that in a moment. So in the simplest part of this paper, I'm not going to go through all of it, it was published last year, but the simple reductionist part is taking NK cells, activating them with cytokines, and then looking at transcriptomes, epigenomes, and trying to understand how these changes relate to one another. So really what Hanyu focused this paper on, in a way, were active enhancers looking at P300 deposition. And what you see is that if you take resting cells, they have plenty of active enhancers, but the active enhancer landscape changes quite a bit, even in a short period of time, in the 6-hour stimulation. And what you do is you gain and you lose these active enhancers, and these are shown here. So you have the acquisition of P300 and the loss of P300 here. And what she wanted to do then is to ask, well, how does this relate to accessibility? So this is sort of the conventional part, this class 3 group of enhancers, where you have deposition of P300, and it's in regions that are already accessible as determined by ATAC-seq. But you have these other regions as well, where you have de novo accessibility, and that's shown here. And again, this is not, we're not the first people to say this. Giacchino Natoli also had showed this in a paper several years ago. We have a different sort of take on this. I'll show you in a moment. And if you compare this with other indicators of enhancers, both poised enhancers and then active enhancers shown here, what you see is this is sort of what we were expecting to see, that this deposition of P300 in these regions where they have poised enhancers, and you have increased K27 acetylation. But in addition, you actually have increase in K4 monomethyl in these regions of de novo accessibility, and then of course increased K27 acetylation associated with P300. But one of the things that Hanyu was thinking about is that you have genes that are sort of induced, but they're relatively weakly induced. And then you have these genes that are just massively induced. And in a way, we were thinking about this from Steve Smale's work and thinking, oh, are these, should you consider both pots of genes here the same? What Hanyu found is that in fact, these regions that have de novo accessibility, these regions are associated with these genes that are more massively induced. So we found that to be interesting. And the other thing that Hanyu found is that these regions are also more likely to have super enhancer architecture. So getting back to the comment before about Rick Young's notion of super enhancers being associated with identity, genes associated with identity. We took a list of genes that have been identified through the IMGen consortium. And what we found is that about with activation of the cells, you have a much greater increase in these highly induced genes compared to these cell identity genes. So weakly induced genes, you have higher likelihood of having super enhancer architecture in these highly induced genes. It's about 50-50 in the identity genes, and weakly induced genes is a small increase. So super enhancers are actually massively organized with activation. And what we found in addition is that stats are the major driver of a super enhancer formation. And so then we wanted to see if we could put this all back together and relate that to accessibility. And that's shown here. So again, this is looking at transcription factor motifs using ATAC-seq and Homer to identify motifs. But I'll also tell you, we did ChIP-seq as well. That's in the paper, and I'll just summarize that quickly. So what did we find? So in these class 3 enhancers, right, these are the enhancers that had constitutive accessibility, and you deposited P300 in these regions. And so as you might expect, you have lots of lineage defining transcription factors that are binding in these regions based on motif. You also see signal dependent transcription factors like the stats, but where you really see the difference is in these regions that are gaining accessibility de novo. So again, in these regions, you see this substantial enrichment of signal-dependent transcription factors, prominently stats, but also factors like NF-kappa B and also AP1, et cetera. What we did, oops, trying to get, there we go. We next did ChIP-seq, and what we found, as we saw here, we clearly see little Tibet motifs here. But the surprise was when we did ChIP-seq, we found Tibet binding in these regions of de novo accessibility. But Tibet was not going to its canonical motif site. So just in the interest of time, as I say, since the paper is published, just summarize this. So acute activation induces many new active enhancers. I have reorganization of the super enhancer landscape and de novo accessibility. And highly induced genes, as I showed you, were more likely to be enriched for super enhancer architecture. And these regions had both constitutive and de novo accessibility. And so what we found, like I said, the idea of de novo accessibility, that's not our discovery. That's the work of others. But what we found is that in the conventional view, you would think that lineage defining transcription factors would be binding to their motifs, and they would be allowing the signal dependent transcription factors to occupy these regions in the conventional manner. But what we found, there are many examples in which actually Tibet was bound to sites that actually did not have a Tibet motif. And so in this case, it seemed like the stats really were functioning potentially as pioneering factors, and somehow Tibet was coming along. And exactly what is the mechanism for this, whether it's phase separation or tethering or things we don't know about. But we were very struck by the fact that the total levels of Tibet binding throughout the cell didn't change. Tibet is not increased in the levels of Tibet don't increase, but the distribution of Tibet changes, but it's dependent upon STAT4. And again, we could show that by the appropriate knockouts. So I'm going to stop there and move on and just summarize a couple of other things to stay on this theme of super enhancers and get back to the point that I was making before, that when we were first thinking about, why are there so few active enhancers shared between, as I showed you, with T cells, macrophages, et cetera. And again, the interferon gamma is just a terrific example of that you have this one region that has so many of these active enhancers. And even just in Th1 and Th2 cells, this helps explain that there's these regions that have this tremendous density of enhancers. And the interferon gamma locus is an interesting locus from the point of view, I mentioned before that we found that long non-coding RNAs are associated with this super enhancer architecture. And the interferon gamma gene has an antisense long non-coding RNA shown here. And so another good example of a super enhancer locus with its presumably cognate gene and its long non-coding RNA gene partner. And again, lots to discuss about this. I'm just going to again hit some of the highlights and say I think we know a little bit about how this thing works, but again, mostly we're quite ignorant. And so I'm just going to show you the data. This is one way of showing the interactions between this long non-coding RNA and this locus. We did this with ChiaPet and again, work from Hanyu, but I'm going to show work that Hanyu did using Hi-C in just a moment. But clearly, you see this locus here interacts with this locus. And so what two former members of my lab, Katrina and Francesca, did was to make two knockout mice. One, we made a knockout mouse in the old way. It took us forever to make this mouse, and it was a mouse in which we deleted the first exon and intron of this long non-coding RNA. And then as we were making this mouse, CRISPR-Cas came along, and it was actually pretty easy to make this mouse here, which we put an insertion of a termination sequence so that we were unable to make the transcript of this long non-coding RNA. And ironically, both of the mice were generated more or less at the same time. And what we found was that if you infect mice with Toxoplasma gondii, both of these mice, the mice that either had the alteration of this long non-coding RNA architecture or just a failure to produce the transcript, both mice were susceptible. But the mice in which you had the architectural alteration were more susceptible and had more cyst burden. Again, papers been published, so I'm going to again hit the high points here. Probably the biggest surprise that we found with working on this long non-coding RNA was this experiment. I was presenting this to Rafi Ahmed a few years ago, telling him about this long non-coding RNA, and Rafi pointed out to me that they did this unbelievably heroic experiment in a way. They vaccinated individuals with yellow fever vaccine, measured their primary response, and then followed these patients for more than a decade to look at human memory responses. And again, just to hit the high points, what we found is that certainly when you infect people or vaccinate people, you see a primary response in which you see this long non-coding RNA, and with that you see increased interferon gamma. But here, with subjects who have been followed for up to 10 to 14 years, you still see elevation of this long non-coding RNA. And so the extent to which long non-coding RNAs like this can give are associated with immunological memory, we certainly don't know, but it's rather provocative to think about this. And I should say at this time, if we look at interferon gamma itself in the memory cells, you don't have expression. And now I think I'll just summarize this here. So what we did was, this is the a cartoon form of the organization of the interferon gamma locus. I'll show you the high C version of this in a moment. And you have these key elements associating so that you have optimal interferon gamma production. But even if you eliminate the transcription of this long non-coding RNA, you actually you see reduced survival, but you still have enough host defense that cyst burden in the brain is normal. But if you eliminate the architecture of this locus, this is where you really get in trouble, is you have reduced survival and then increased chronicity of disease. Anyway, lots of things to do with this long non-coding RNA, but I just want to do very quickly give a shout out to Hanyu, who gave a talk at the AAI meeting, and I think if it's still archived, and you're interested, it's worth taking a look at this. And I just wanted to remind you that the interferon gamma, like many cytokine loci, is in this multi-cytokine cluster. So you have the long non-coding RNA here, you have the cytokine here, you have a cytokine that's a pseudogene in mice, but in humans is IL-26. And then you have IL-22. And of course, we associate IL-22 with ILC3s as opposed to NK cells and Th1 cells, ILC1 cells, which make interferon gamma. So it's a really interesting locus from the point of view of how you discretely regulate these particular cytokine genes. And so I just wanted to, again, cover a lot of information quickly. I apologize. But just show you here that when you look at the organization of this locus, you have this rather large TAD and sub-TADs underneath this big TAD shown here. And then what happens when you activate cells, you have relative enrichment of some of these regions. And what we noticed here was this one CTCF binding site, which again, I showed you before, is a region where you have interaction between this long non-coding RNA, the locus, and this other region. And it's sort of at the peak of these interactions shown here. So Hanyu deleted this CTCF binding region, and again, the work was presented at AAI. And what she found was that what happens when you delete this key site in the interferon gamma locus, it disrupts the normal chromatin structure shown here. And what we found is that this impairs T-bet induction and production of interferon gamma in Th1 cells, but it's relatively dispensable for interferon gamma production in NK cells and memory CD8 cells. So this is exactly what we were hoping, is that the region have all this complexity in these loci, is that some of these things are cell specific and activation dependent. And what Hanyu then found was that when you had this impaired response in Th1 cells, that's associated with an altered a type 3 response to Toxoplasma gondii infection. So then you have more of an IL-17 response. All right. In the next few minutes, I just wanted to touch on two other quick stories that relate to super enhancer loci. This is shown here. So here's another example of a super enhancer portion of the genome. And in this case, there are two genes, miR221, 222. And these are microRNAs, but they too have this super enhancer organization. miR221, 222 are on the X chromosome, overexpressed in cancers, but the way we became excited by this cluster is that they're preferentially expressed in Th17 cells and also in Th1 cells and regulated by stats shown here. And what we found, I apologize, this is busy, but what you can see is that in the relevant cells, that if you have STAT4 knockout, you have reduction in production of miR221. And in Th17 cells with a STAT3 knockout, you see reduction in production of miR221. And you have binding of the stats to this region. So for all these reasons, we thought this might be an interesting locus to target, which we did. And what we found is that the knockout is viable and fertile. We didn't see any gross alterations of hematopoiesis, et cetera, and grossly T cell development was normal. But what we did see is that we saw, to our surprise, but also delight, was that you actually see more IL-17 production and more ROR gamma in the knockout in the intestine, and that's enumerated here. You see increased IL-17, increased ROR gamma. And when we challenged these mice, what we found is that we saw increased pathology in the knockout mice, and that's illustrated here by loss of weight, and also increased cell infiltrating the intestines of the knockout mice, and again, that's enumerated here. Now, the reviewers appropriately pointed out to us that that this didn't really tell you anything about the T cells per se, since it was a germline knockout. So we then made CD4 knockout mice, and fortunately we found the same phenotype, that you have increased pathology when you delete miR221, 222 in these mice. So the next question, of course, is, well, what are miR221, 222 regulating? And so we looked at potential targets and potential genes involved in the Th17 signature, and we were relieved to see that there were some candidates here that in fact are regulated by miR221, 222 and participate in Th17 cells. And a particularly attractive one to us was IL-23 receptor, but also MAF. But you could see by making constructs and seeing whether these microRNAs regulate the constructs that have the miR221 site. You can see that the IL-23 is repressed by the microRNA, and so is the MAF construct repressed as well. So then we went back to the mice and we checked to see, do you see alteration in IL-23 receptor signaling? And that is the case, that you see in the knockout, you see increased responsiveness to IL-23, shown here as measured by both IL-17 and RR gamma. And again, that's quantitated here. So to summarize this, IL-6, 12, 23, acting via STAT3 and STAT4, regulate this microRNA. I didn't show you, but it's interesting that TGF beta actually negatively regulates miR-221, 222. And the extent to which this contributes to this idea of homeostatic Th17 cells versus pathogenic T cells, that it's a possibility that this is one way of thinking about that. IL-23 receptor and MAF are miR-221, 222 targets. And in the absence of miR-221, 222, you have the IL-23 receptor because IL-23 receptor is a MAF target, and you have increased susceptibility of DSS. Okay, I have 10 minutes left, and I just wanted to touch on another quick story. I think I can do it, but it illustrates again, as I hope I made clear, that we're sort of in love with super enhancers, that every time we find a locus in the genome that has a super enhancer, we take that a little more seriously. And this is another example, and this is an enzyme, cholesterol 25 hydroxylase, which again has a lot of work done by Jason's sister and others on this, not so much in T cells, but in macrophages. But this was an interesting locus to us because, once again, it has super enhancer architecture. And it also actually has two antisense long non-coding RNAs. So I think that's going to be my rule when the next postdoc comes and says, I think I want to write a paper on a gene. I think the first thing I'm going to say, does it have a super enhancer architecture? Does it have long non-coding RNAs? Does it have STAT binding sites? If not, I'm not interested. With this business about trying to think about this sort of exquisite aspects of regulation, that's it's sort of what I think super enhancers are indicative of. In any case, others, not us, before us, have found that interferons and IL-27 induce cholesterol 25 hydroxylase. And this was interesting to us because IL-27, we've sort of been involved in IL-27 for many years, including Alejandro Villarino's work, starting with Chris Hunter's lab. IL-27 promotes Th1 responses, but has essential regulatory functions. And so it can induce interferon gamma, but actually what it's most important for is its regulatory functions. So we were intrigued by that. And we found, in fact, that IL-27, along with interferons, induce CH25H. It's interesting. It's not rapidly induced. It's delayed in its induction, and then its induction is transient. And then you see the product 25 hydroxycholesterol in the supernate. And so you see the gene go up, and then the cholesterol the hydroxyl form cholesterol in the supernate. And interestingly, and I think we have a bit of an explanation for that, is that it's not induced in CD8 cells. It's induced in activated naive and memory cells in this way. But I'll show you a little bit more about that in a minute. And so this was the part that really got us interested, is that when we stimulated the cells with IL-27, you could certainly see that in the absence of TGF beta, you see induction here, and the wild types shown in the dark bars here, that you see IL-27 induces CH25H. But to get maximal expression of CH25H, the combination with TGF beta gives you much more impressive induction. And so this was interesting because when we had used Tibet knockout mice, we found that you actually then didn't need the TGF beta. You got very high level induction of CH25H when Tibet was lacking. And the other thing, of course, is that the converse is that one of the things that TGF beta does is it represses Tibet. And so, and then we obviously looked at the locus, at the super enhancer locus, and you can see once again that you have the locus is more active here as measured by K27 in Tibet knockouts. And there's this enhancer that's quite distal from the gene itself. And so this is sort of the model that we're thinking about, is that as I was saying, what's the difference between IL-27 and interferon gamma? Well, they both act on STAT1, and so they both have the capacity to induce CH25H. But if you highly induce TBET, that's going to counteract the effect of the cytokine. And so in that case, that's why this combination of IL-27 TGF-beta give you this nice induction of CH25H. So what does CH25H do? Again, this is not our work. It's 25-hydroxycholesterol actually inhibits many aspects of cholesterol metabolism, shown here, and that's what we found in our T cells, although Jason Sister and others have shown this previously. And so as a result, what 25-hydroxycholesterol does is it basically blocks proliferation and increases the cell death in T cells. So you have impaired viability when you add this form of cholesterol, but you can actually add excess cholesterol that reverses the cell death that 25-hydroxycholesterol induces. Now, the other thing that's interesting about this is there's this transient effect of 25-hydroxycholesterol, that you have a big effect early on, actually in cells that are not making high levels of CH25H. So you have greater susceptibility in early activation, and later the cells become resistant to the effects of 25H. And it took us a while we sort of puzzled through this, but we did a mixing experiment, which is in our lab is usually the way you sort of sort these things out. And so it's a bit of a complicated experiment, but and it has lots of different controls in here. But what was done here is with cells that either have wild type cells that don't have the enzyme CH25H, and then you stimulate with IL-27 and TGF-beta, generate these cells, and then you remove the effector cells and then add potentially targets that either have IL-20 receptor, 27 receptor or not. And then you treat these cells, we put TGF-beta antibody in to block the effective TGF-beta. And with that long, complicated sort of description, what you see is that, in fact, the effect, we think, is more paracrine. So the paradox is that when the cells are maximally making the enzyme that makes this product, they're now resistant to the effect of this, but its partners that are not being activated at the same time are the ones that are going to be sensitive to this cholesterol product. So you see maximal induction of the enzyme late, maximal sensitivity to this product early on. And so we would argue that the main effect of 25-OH hydroxycholesterol is paracrine, not autocrine. So does it make a difference in disease? And so we use this particular model of pemphigus. This is disease induced by desmoglein 3 specific T cells, which our collaborators have generated. And then what we did this in an adoptive transfer model that I'll show you in a moment, but we did it in other models as well. But just to show you here, if you take mice, these are normal mice shown here, you adoptively transfer the pathologic T cells, you see pathology here, and actually then if you do this in with cells that don't have 25 CH25H, you can see much more infiltration of cells in these mice. But we also confirm this with other models, immunization of the transgenic mice, and also in a hypersensitivity model. Okay, so to summarize this, IL-27 TGF-beta induced CH25H and CD4 T cells, TBET inhibits this, it impairs the ability of interferon gamma to induce CH25H, since interferon gamma is such a good inducer of TBET. TGF-beta inhibits TBET expression. We also think this is probably one of the reasons why CD8s don't do this, because they have EMAs, et cetera. CH25H induction is delayed and transient, and so the net effect is the effect of 25-hydroxycholesterol. While it inhibits cell proliferation and promotes cell death, it does so in a paracrine manner, and in that way, this is the way that these cells act as regulatory cells to limit T-cell mediated pathology. Okay, so getting back to what I said before is that, in the 90s, we were thinking, we just wanted to know the players. How does this get to this? And the JAK-STAT pathway was elucidated. But then when we discovered the complexity of the genome, we realized that the work was just beginning to understand, how on earth STATs acted throughout the genome on the paucity of genes that we have and non-coding region. And again, what's so fun about all this is that, you really do see things differently as you get new tools and new capacity to understand the genomic response to cytokines, and not just thinking about focusing on promoters. And the thing that I hope with all of that is, I think, we just in the early, in the 20th century, we developed biologics, and they're amazing drugs. They're terrific for treating patients with all sorts of autoimmune disease. But now we have JAK inhibitors, and the good news with JAK inhibitors is it's sort of like a rheostat. We can titrate the doses more easily. They have short half-lives. If you have a COVID patient who's not doing well on JAK inhibitors, you can stop it, and the drugs are gone quickly. But I do think, if you think about the fact that in a T cell, there's 20 some odd thousand switches, dials that we can be manipulating, it really would be nice if we could benefit from that knowledge, and then treat patients by taking all this information into account as we adjust immunotherapy and different agents. And I think I mentioned people's names along the way, and I'll stop there, and I'm 2 minutes over. I apologize.
Well, thank you, John, for the amazing seminar. This is a really great example of how to deliver a wonderful speech. I think a really good lesson for all of our trainees. Really, thank you. So I think with that, let's open the floor for questions. Chris, do you want to get started? Let's see, we don't have anyone. I see Li Fang has a raised hand. Let's start with Chris, and then Li Fang, and then Caitlin.
Okay, John, can you hear me?
Yep.
Great. Okay. Yeah, I wish we had another hour.
Yeah.
there are lots of things we could talk about on the super enhancer and lineage determining and signal dependent front. And I think I will get back to you at some point on that. But the question I thought I would ask has to do with the very last component on the 25-hydroxycholesterol. And just one thought, I mean, that's also considered to be a ligand for LXRs. And I did see, I think, an LXR target gene that was up in addition to the SRABP genes that were down on that little gene expression plot you showed. I'm very curious as to whether paracrine signaling through 25-hydroxycholesterol could be regulating the liver X receptors in other immune cells and epithelium and so on in the skin. There is some evidence that LXRs do actually play biological roles in maintaining integrity of the epithelium and so on. So have you looked into that at all?
We have not. Yeah, I mean, it is one of these areas that I'm trying to educate myself on lipids, et cetera. And yes, there's certainly, just reading some papers on LXR and how that influences, lipid order and how that may influence signaling via cytokine receptor or T cell receptors. But no, we haven't done that. But I need to read these papers.
Yeah, I think that would be. I think that there may be an angle to that, as well as the suppression of cholesterol biosynthesis, which obviously does occur and is a big part of what's been shown, but the LXR angle could be interesting.
Yeah, and I should say all that stuff about lineage defining transcription factors. I stole all of that from your reviews. I should have made that clear from the beginning.
Yeah, no, thanks for the shout out. Yeah.
And Li Fang?
Hi, John, that's a great talk. I also want to focus on the last part of your talk about the 25 cholesterol hydroxylase. So you start from saying both interferon gamma and IL-27 can induce it. And then obviously you emphasize IL-27 play more immune regulatory role as what we know so far compared to interferon gamma. But the data you show later on didn't really separate the effect of interferon gamma and IL-27 because both of them induce TBET and both of them can induce 25 cholesterol hydroxylase. So I would imagine that if you do interferon gamma plus TGF beta, you should get the same effect of just like IL-27 plus interferon TGF beta. So what really make IL-27 different here? That's my first question. And the second question is, so TGF beta plus IL-27 has been shown by Daniel Masita that play an important role to promote CD8 alpha alpha CD4 IELs in the gut epithelium. So have you, because you said that CD4 produce 25 cholesterol hydroxylase but not CD8, but in the gut they become CD8 positive, CD8 alpha alpha positive. So have you looked at those population and do they produce 25 cholesterol hydroxylase and whether that also play a role in the gut? Thank you.
Great questions. To answer the second question, no, we haven't done that, but I think, Ananda was at the NIH last week and kept reminding us by all her beautiful work, focus on different responses in tissues and that the tissue signals are important aspects of gene expression. Let me just go back for what I think I showed you, and maybe I won't waste your time. I think what the data showed that I was going through very quickly is interferon gamma is a relatively poor inducer of CH25H. And we think the reason that is it's such a good inducer of TBET. Now, if you got the secret sauce of getting the right dose of gamma and the right dose of TGF beta, I think you probably would be able to induce CH25H. Type 1 interferons have been the inducers of CH25H for a long time. And again, relatively speaking, type 1 interferons poorer inducer of TBET than interferon gammas. So that's what I meant to say, although it's possible it didn't come out of my mouth accurately. Does that make sense to you?
Sure. So it's basically the level of TBET that can be induced by different cytokines that potentially dictate the impact of those cytokines on the induction of 25CH.
That's what we think. Yeah, that's what we think is the key thing with this phenomenon.
Okay, thank you.
Sure.
Good. Caitlin?
Yeah, hi. Thanks, John. I was going to ask sort of similarly to everyone else about that last part of your lecture. It was really exciting. So I think it's interesting that the cells that produce CH25 are not as affected as the cells around them. And I know this was in vitro that you looked at it, but do you think that, or have you looked into whether either the CD4 or the CD8 T cells are more heavily affected by the cholesterols produced by CH25H active cells? Or like, is there specificity, or does it just promote activity in everything?
No, it's a good question, and the paper is now back for its second review, and what the reviewers made us do is to really dig into this, autocrine versus paracrine effect, which I think we sort of selective, we have an answer to that, but unfortunately we don't have an answer to, I think the question you're asking is, why do cells that are fully activated the way we activate them, why do they become resistant? And is it, is it Chris's idea, is it LXR, or also, right, I think CH25H can bind, an orphan, not an orphan anymore, but the G protein coupled receptor, right? So some of the effects are receptor mediated, and also, as Chris was saying, LXR. So that's a long way of saying I don't know.
Great. Well, let me take the last question. Oh, I see Sue has the raised
Hey,
hand.
Sue.
Sue, go ahead.
Hey, John, great.
How you been? Yeah.
Great to see you.
Yeah.
This may be too disconnected, but also just thinking more about the cholesterol regulation.
Yeah, but I should have skipped the whole front part of the talk and just, okay. Nobody cares about all the rest of this stuff. Okay, go ahead. Yeah.
I wanted to try to connect it back with your work where you showed several years ago about TBET regulating type 1 interferon production in T cells. And because there's this interesting link as well that Steve Benzinger has also found, where if you suppress cholesterol synthesis, you can promote type 1 interferon production in macrophages. And so I'm just wondering if you think that these two processes might be also connected in T cells.
Right, you're saying our paper where we said that loss of TBET allows
Type
the
1
cells, that they misinterpret, right?
to produce type 1 interferon when they're
and you're saying if you delete TBET, you'll have more
CH25, and so therefore would that be
because some of it is sort of the autocrine interferon production.
And is that, and I guess my real question, the heart of my question was, is that linked to the pathology? Is it is some of the pathology that you're seeing type 1 interferon dependent? So do these cells have a type 1 interferon signature as well that lack CH25H?
I think, yes, sadly, I think you read my paper better than I read it, and I need to go back. It
may connect, but I was just trying to think.
Yeah. It's an interesting possibility. No, we didn't. But I'm sure when the paper comes back again, the reviewers will give us another chance to improve the paper. So we'll see. But yeah, good point, Sim.
Great. Let me take the last question, John. I'm really interested in the midsection with the CTCF knockout, the interferon locus. Did you guys look at IL-22 or IL-33 in that animal, and what happened to it?
Yeah, we're in the process of that. We considered a number of possibilities that you would have, that CTCF would be acting as an insulator, which it doesn't really seem to be doing. But so what you should do is next year you should invite Hanyu Shi back, because by then, things have been slowed down a lot by the pandemic, of course. So there are a bunch of experiments we'd like to do with those mice, and it's hard to do. But yeah, that's exactly what we were getting at. Was it's to me, it's just such a fascinating locus, and why you turn on these two genes, in separate contexts. We didn't see that in the simplest version of the experiments, but it's something we ought to do. But Hanyu would be the best person to tell you that part of the story.
Awesome. Well, great. Well, join me to thank John O'Shea for a really wonderful seminar. And thank you so much for all of you for coming and attending our last seminar of the year for the immunology program. Super excited. I'm sure we're going to have a fantastic program again next year. So John, thank you for being our best and last seminar speaker for the year. So thank you so much.
All right. Bye-bye.